Inflammatix was founded to focus on bringing precision medicine to the point of care, and our first targets are in acute infections. So my training actually was as a general surgeon. I had a clinical background, and I was always very frustrated with the state of diagnostics for acute infections because nothing fits into workflow, and the tools that we do have that are too slow anyway also sort of don't answer the questions we need to answer. And so we tried to solve a big problem, which was whether your kid is homesick and you're trying to figure out where they need antibiotics, or you're showing up in the emergency department feeling like you might need hospitalization, or you're a patient in the ICU, you need an answer in minutes. And so we tried to start there. What if we could create novel diagnostics that fit into workflow? Well, the problem with existing diagnostics is not just that they're too slow. The biggest problem is that they don't work. If you sort of look at your right side of the screen, there's a lot of froth in the space of clinical diagnostics for sepsis, and most of that focuses on diagnostics to detect bloodstream infections better. The fundamental problem is that most infections aren't bloodstream infections. In cases of sepsis, only about 40% of patients actually have a bloodstream infection. Most are actually locally held in like the skin and soft tissues or the lungs or the belly. And if you go into a broader population, something like emergency department patients that are just diagnosed with an infection at the end of their hospital stay, only about 10% have a bacterial infection. And outpatient pneumonia, it's like 2%. So you have this big diagnostic gap between sort of where most diagnostics focus, which is looking at the blood because it's easily accessible, and what physicians want, which is something that can tell you whether or not you have an infection, not just a bloodstream infection. So I trained as a physician, but we also went out and started talking to emergency department physicians and said, what would be the ideal diagnostic? What are the key questions you're trying to answer when you have somebody come in who's, acutely in extremis? And they gave us these answers. Number one, do I need to prescribe antibiotics? That's currently essentially a total guess at the time, when you see a physician and they give antibiotics, they're basically just guessing. Then what other tests does the patient need, and where are they going? Are they coming to the hospital? Are they safe to go home, et cetera? And if you can give a test that's accurate and that in particular turns around in less than about 30 minutes, that would be a practice change in the way these patients are treated across the hospital system. So Inflammatix's approach is a little bit different. We're diagnosing infections, but not by looking for pathogens at all. What Inflammatix does is we read patterns in the immune response, mRNA patterns in the bloodstream from white blood cells. And by doing so, we can detect an infection anywhere in the body. So not just bloodstream infections, but the circulating immune response will tell you if there's an infection in the lungs or the belly or the soft tissues. We can differentiate really accurately between bacterial and viral infections. We can risk stratify for the risks the patient may decompensate into sepsis and need ICU care. We can predict therapy response. And because this material is really abundant, and I'll get into this a little bit later, we can do this really, really quickly. We're not searching for ultra rare events in the bloodstream. So the first product, and the sort of case in point I'll tell you about today in the machine learning section, we call HostDX Sepsis, the HostDX product pipeline. HostDX Sepsis is a single test that gives you 3 scores every time you order it. So the first is, what's the likelihood that this patient has a bacterial infection? The second is, what's the likelihood that this patient has a viral infection? Meaning if you were co-infected, you could be high viral and bacterial. Or if you're suffering from a non-infectious cause of injury, you'd be low. And then the third is an estimate of severity. How likely is this patient to need ICU care or progress to mortality in the next 30 days? And then each score is sort of broken into these highly actionable bands, and you can see that for most patients, we can place someone into a sort of sensitivity and specificity well above 90%. So it's far more accurate than prior tests in the space. And I'll put the sort of the punchline right up here at the front. The test works really well. So what you're seeing here is results of the first version of our algorithm for identifying the presence of bacterial infections. And the top line in gray there showed the sort of the training accuracy and AUC of 0.91 across 18 different clinical cohorts. But so far in 3 blinded external validation cohorts, we've maintained that level of accuracy. So you see our machine learning models suffer from no overfitting, even through sort of transfer onto rapid molecular platforms. And this level of sensitivity can support this really, really high level of sensitivity and specificity for the vast majority of patients. compared to clinical standard of care, which is currently procalcitonin, you can see there's a major boost in the accuracy there. So that's what we're doing, and obviously the question here is how. So take you a little bit back into your immunology days, for those of you that took it. Patterns of mRNA in the cells essentially tell a story that we can read at Inflammatix to produce clinically actionable insights. So on the left-hand side, you see sort of different stimuli into the body cause different signaling patterns downstream in the immune system. And we're primarily looking at the innate immune system, neutrophils, macrophages, things like that. So you can see maybe one type of bacteria, gram-negative bacteria, might sort of kick off one downstream signaling pathway. Gram-positive bacteria, same general genes downstream, different pattern of activation, non-infectious, viral inflammation. So we know sort of it's the same genes that are generally involved, they're just activated to different levels of degree. And what HostDX Sepsis fundamentally does is we've pre-selected variables, genes, that we know are highly involved in this, and I'll tell you about that in a minute. And then, of course, we use machine learning to look at the pattern of activation to basically figure out what it was that activated this particular immune response in the first place, and that's what we report to clinicians. Seems simple, but people have been trying this for about 2 decades, and they've always failed. It turns out that this is a really, really hard space. Not only do you have all the traditional sources of heterogeneity, age, gender, severity, whatever, there are at least 3 sort of time courses that are massively different in patients who show up with acute infections. So you see them on screen. On the x-axis, sort of time since infection, right? If you show up in the hospital 2 days after feeling sick versus 7 days after feeling sick, your immune response looks a lot different. On the y-axis, time of sampling. Every one of these studies involves some amount of leeway for a clinical nurse to go gather a sample. And maybe it's 2 hours after the patient showed up, maybe it's 6 hours, maybe it's 24 hours after the patient showed up in the hospital. And then the last is, especially for hospital-acquired infections, people come to the hospital for something, surgery, a car accident, a heart attack, whatever, and they develop an infection during the time there. And so you're looking at sort of an immune response due to a hospital-acquired infection on top of the background of whatever brought them there in the first place. And the reality is that almost every study that's been done, including ours, almost always looks at a really small subsection of these sort of sources of heterogeneity, right? So we'll enroll something that looks at like just community-acquired pneumonia within 24 hours of admission, or just trauma patients that have hospital-acquired infections occur. Because you can't just sample everyone in the hospital all the time, because the problem is we actually need physicians to go through every single one of those charts that we gather and label the patient as having a bacterial infection or not. So on top of this, of course, for the clinical diagnostic to be worthwhile, it needs to be generalizable. So it's not just that there's this really broad space, it's that we need to find sort of some kind of complex hyperplane, right? Something in 29 dimensions, since we use 29 mRNAs for this test, that can separate always the bacterial infections from the not sort of across all of these dimensions. And that's really the challenge, right? Is coming up with something where we're not massively overfit to our training data. Unfortunately, that's pretty much industry standard in this area. Essentially, every academic group does what you'll see on screen, which is pre-selects some genes and then goes to a new cohort and says, well, we relearned the model in this cohort, but see, trust us, the genes themselves were informative. And so we cross-validated locally and we have high AUCs. Of course, for the data scientists and machine learning experts of you, that's somewhat of a laughable approach, but it also means that you've never actually created a diagnostic that would be forward applicable to a single patient, right? You've never locked one version of a diagnostic and applied it in a blinded fashion. The historic problem, the reason people wouldn't do this, is there was never a platform that was high multiplex that would also be rapid, right? So typically, for instance, you'd use microarrays, or now more recently RNA-seq, but those are sort of too slow. And so often what you get is somebody selecting a bunch of genes from microarray studies, transitioning into a PCR-based platform, and then applying. And it's the difference between the platforms is why they say, well, we couldn't apply the prior signature onto the new platform. So not surprisingly, Inflammatix does something a little bit different, which is we've pre-selected some genes. We learn an algorithm across technical platforms, and I'll tell you about that in a minute. And what that means is that we can now start doing blinded studies showing that the same diagnostic maintains its accuracy across multiple patient populations. And this pre-validated model is now sort of ready for FDA clearance. So there's two parts of this that are computational. The first is trying to select the variables themselves. And this is actually largely work that we've published on very extensively out of Stanford, which was essentially a machine learning free model, non-parametric. Just can we find genes, sets of genes that are highly differentially expressed in a condition of interest. And so I told you, what we want to do is represent the heterogeneity of disease, right? Old, young, men, women, inpatients, outpatients from all around the world. And the way we did that is we first did a massive search of the public domain. So we trawled sort of all of the extant data sets and found all of those that would look at, for instance, patients with infectious and non-infectious inflammation in the same cohort. And we put them all together at once, and we perform essentially a meta-analysis and then a non-convex optimization to find which of those gene sets are likely to be highly diagnostic. We published a few papers, one showing 11 gene set for separating infectious from non-infectious inflammation, one showing a 7 gene set for bacterial versus viral, one showing another 11 gene set for 30-day mortality prediction. And the sum of those, 11 plus 7 plus 11 genes, that's the 29 mRNAs that now make up the core of HostDX Sepsis. And that actually is where Inflammatix's story begins. So what we do at the company now is we say, okay, we've had some variables that we believe strongly are highly diagnostic. And what we want to do is learn a stable machine learning model across all those things. Now there's two sort of insights that we use in order to do that. One is a very large proprietary database. So from the, well, I'll say very large is a relative scale in this room. In the public data, it's maybe about 1,000 patients. Now at Inflammatix, we've upped that to around 3,500 patients, and our database is roughly doubling every year. We use carefully physician phenotype data. So as I said, we know that sort of blood culture alone isn't enough to get you to figure out who has a bacterial infection, who doesn't, right? That's the problem we're trying to solve. So every single one of our data points is actually hand labeled by 3 physicians as being bacterial, viral, or something else. And that's FDA's standard as well. So curating the data takes a long time. We then use a couple of proprietary tools, one in data normalization to bring together data, for instance, from an AFI platform, an Illumina platform, and RNA-seq, knit those together into one sort of stable matrix, and then try to learn one stable machine learning algorithm across all of that. And then, of course, we have to transfer that into a rapid proprietary device. Now, I showed you the data already, so I'll just say the proof is in the pudding, but the question is, arguably, how do we do this? Unlike many in the room, dealing with trillions and trillions of data points, ours is a problem of small data. We have thousands of samples, but they're spread across dozens of studies. You almost can't find a center that will do more than 100 or 200 patients because the heavy lift on the physician side is just prohibitive. Nobody wants to do all of the hand phenotyping. And so what we find is, our data looks something like this, right? There's a T-SNE of first a bunch of data points colored by study, and you can see it's very, very heterogeneous. And on the sort of in the middle, data points colored by class. There the blue is bacterial, the red is viral, the yellow non-infectious. And our goal is not just to learn something that sort of works across all of these domains to separate these patients in machine learning space, but then also, of course, to find something that will be transferable onto a rapid platform that has its own sources of noise, because these are biological measurements, right? We're not perfect at it. There are, if you're trying to measure gene levels within 30 minutes, always challenges with making sure that each level of accuracy is correct. From a machine learning perspective, we've actually developed an in-house customized pipeline we call Inflammatix Machine Learning, IML. Represents all of the sort of standard model types you'd imagine, anything from penalized LR with a single hyperparameter all the way out to sort of extensible artificial neural networks with dozens of hyperparameters. And the software itself sort of allows for advanced cross-validation, and in particular what we call a leave one study out validation, which I don't have time to go into, but happy to talk about offline. We can do both random and grid searches, and certainly it's sort of flexible for model selection downstream. And this is the primary use for us, at least, of AWS, as we have a single sort of bash callable line that will split this out into AWS, and we use AWS pretty extensively during novel hyperparameter searches, although certainly we'll do on-prem and local searches once we've sort of answered the big questions, and now we're just fine-tuning or answering smaller sort of model optima questions. The structure itself should be fairly straightforward for any of you, right? Simple sort of map reduce based on Hadoop, sent out, and then regathered into a pickle file. Why use the cloud? So just to give you a sense, right? Here are 10, a few at least, hyperparameters that you would need to optimize over just for an MLP. You can generate tens of millions or billions of possible models out of the hyperparameter search. And I pulled the numbers, so we typically do 100,000 hyperparameter search, which for us takes about 12 hours on 40 AWS C large instances. And for us, that basically means that our machine learning engineers can begin to hypothesize interesting questions about novel architectures and novel techniques, and not sort of be unduly waiting for just the numbers to crunch. So fundamentally, this is what we found: is Onscreen is something we call a spoke net. It details basically are that we found that individual gene models, if we put those into the structure of a neural network, we sort of pre-select what we know is going to work together. It turns out to build a much more generalizable network than just throwing all the data in and saying, go find whatever works based on a random search. But those kinds of insights, you have to test them, and there's no sort of automated way to test for, should we put the severity genes sideways above the diagnostic genes or underneath? It takes biological insight when you're building these things, and so each time it's a new search, and for us that means we sort of have to be on grid. And then of course, when we move to things like Bayesian optimization or whatever, all of those each time it's a massive new search. So for us, the ability to rapidly scale up during periods of great need has been a game changer. Overall, then, I'll say the key learnings from this particular project in HostDX Sepsis have been sort of twofold. In general, what we've shown is that proprietary data and clever data science or machine learning essentially allow us to solve previously unsolvable problems. So, the next closest groups that are trying to do something like this typically get worse than random results if they try in external blinded data. They publish that. We're the first to show that we can actually find something that's stable in external data, and it's largely because we've brought in domain expertise on both the medicine and biology and the machine learning sides. And I think what we've come to appreciate is that you can't just take a machine learning engineer that's been building better ad engines for four years and sort of put them directly onto biodata. But similarly, the people that know the medicine or the biology have to be able to speak data science a little bit in order for those two groups to come together and produce new insights. And in general, I'll just say that the small data domain has its own unique challenges and opportunities. This is small enough that we can't just rely on the unreasonable effectiveness of data. You can't just put it in and assume that some big convolutional net will work. There has to be insights, but we have shown also that those insights can lead to substantial gains in performance. In terms of the cloud, we use it, as I said, for on-demand scalable compute to sort of allow for novel hypothesis testing primarily. We don't use convolutional nets, and so we found that the CPUs work fine. We don't use GPUs. I'll mention just a couple of learnings for us have been putting alarms in and stopping instances when not in use. We've gotten occasionally, with due respect, some bills that we were a little bit surprised by. The other thing is that initially early on when we were doing sort of very large searches up to 1,000 nodes at once, we found that we were actually subject to throttling, and a quick phone call took care of that. But for those that are starting off early, there shouldn't be an issue. If you find an issue, we found Elliot and Paul to be very, very helpful in making sure that the very large searches suddenly happen very quickly. So what's next for us? This is true for sepsis and sort of bacterial infections, but the immune system in general is a universal diagnostic. The dendrogram on the right came from a paper that we published out of Stanford showing that essentially a gene signature of no more than 100 genes is sort of trivially able to separate any two disease states. And it's interesting scientifically. Of course, as a small business, we want to move to places where there's actually value to be created. But the point is, we can do this a lot of times. And so Inflammatix is now building a pipeline of tests, at first focused on sort of acute infections, both in the hospital and outpatient setting, better tests for influenza, but then moving into things like companion diagnostics, whether for sepsis immunotherapies, TNF-alpha response, NASH, et cetera. Have gathered some partnerships to date. I'll simply mention that if those of you on the therapeutic side are interested in custom companion diagnostic development, happy to talk at the break. In general, all of this goes on to a rapid multiplex point-of-care system, so we can sort of deliver these signatures either via finger stick at the point of care, not today, but within a couple of years, or much more standardly via venipuncture in a hospital setting or clinic to do sort of multi-gene signatures in less than 30 minutes. And so for us, we feel that Inflammatix is sort of uniquely poised now to bring together the two great areas of diagnostic innovation, which are sort of taking existing tests and moving them to the point of care where a lot of value has been created on the y-axis, but also certainly the sort of precision medicine approach to novel gene signatures where clearly value has been created, and yet every single one of those takes 7, 12, 15 days to turn around, right? So if you have a 12-day turnaround sepsis test, it could be as accurate as you want it, but sort of nobody cares. What you need is something that fits into workflow, and so that's where we're trying to marry these two. So with that, I'll take any questions you have. Thanks very much.